and Dr. P.R. Raghavan, we like to call him Raghu. He's endeared that way. And basically, he's the chairman and CEO of Arnox Incorporated. Raghu has a PhD in organic chemistry from Oregon State University in 1979, and MS in chemistry in 1972 from IIT, Mumbai, India. He has worked on drug discovery for over 25 years at Columbia University, Max Planck Institute, Germany, Novartis and Boehringer Ingelheim Pharmaceuticals. His fields of specializations are CNS, inflammation, AIDS, and ACE inhibitors. His experience in drug research includes chiral synthesis, process, scale-up, and clinical research. He has been working for the last 15 years in the area of nanomedicines and food supplements.
If you work on the assumption that whatever is accepted as true, then there will be no hope of any advancement in science. So the major problem in the world are the result of the difference between how nature works and how people think. The problem is we don't look at nature. We are just looking at how people are thinking. All my work you can download from the site, researchgate.net or academia.edu. You will find all of them. One of my patents, the one in 2014, the notice of allowance from the date it was published to the date it was allowed was exactly 31 days, maybe less. And this was the fastest patent approval in US history since 1791. See, all the work that I'm talking about, we do this by third parties. So every work I report, we have outsourced it to people who do it as a service contract with us. So, for example, LSU Health, which we used, they did all the rat studies. The Virginia Tech, we used, we outsourced all our gene expression work to them. They did it. The Utah State University did the viral research for me. So we're all over the world. And this is the problem, which was forecast in the 50s, that medical science has made such progress there's hardly a healthy human being left. And, there's an old Ayurvedic proverb: when the diet is wrong, medicine is of no use. When the diet is correct, the medicine is of no need. So today we have more experts, more drugs, and less wellness. And one of the mistakes that people have made is that we assume that all modern diseases are unrelated conditions and don't have a common cause. That's why, you have experts in endocrinology, cardiology expert, you have an expert in arthritis, you name it. We have broken the human body up into so many tiny pieces that there's an expert for every little piece. But actually, all of them are related. And you see, disease is a cellular phenomenon. So we started with a single cell when life evolved. And so every disease is reducible to the disturbance of cells. And therefore, it was said by E.B. Wilson, the Harvard biologist. He said the key to every biological reaction is in the cell. And by that, they mean the nuclear receptors. And the other thing we have to notice is that our cells, 90% of our cells are bacteria, viruses, the other microbes. 7% is junk DNA, and only 3%, about 23,000 genes, are actually what is the focus of today's pharma and medicine. We ignore the elephant in the room, which is the human microbiome. We are ignoring the most important thing, which happens to be the microbiome. So we are only 3% human. So basically, we are about 10 trillion cells we have, human cells, but we have 100 or even more trillions of cells, which are bacteria and viruses. So if you look at our bacterial information, 99% is bacterial and virus genetic information that we carry. And this is the other thing that people in this world have forgotten. We are now shut down the world for one virus. But look at the number of viruses we have in the world. It's 10 followed by 33 zeros. That's just the viruses. Then you have bacteria, then you have other things. So we only 10 to the power of 9 human beings. So we are a minority in this world, And the only way you're going to survive against this kind of numbers is you need to protect yourself with more immunity, to protect yourself from these guys. Most of the bacteria that we have is actually in the gut. And, people don't understand that all these medicines that we take, they disturb the gut bacteria. This was published, 10 years ago, Cornell University, where this is our bacterial diversity on the left, the different colors. Look at it after 7 days. You took a course of, clindamycin, which happens to be an antibiotic. It takes 9 months before you begin to get some kind of diversity. All your bacteria is destroyed. And this is the problem with each and every drug. You are destroying your gut bacteria. People think it's fun just to take a pill for every ill. So you need to do it in a way that doesn't harm your gut bacteria. And this is the classical problem which didn't exist until 2000, which was when Craig Venter sequenced his own genes, the first human genome. What we have found since then is we only have 20 to 23,000 genes, but for every gene, there are 360 microbial genes. So that adds up to, if you multiply 20,000 by 360, you get 7.2 million genes. So if you want to target a gene, which is what all the pharmaceutical companies talk about, it's 1 in 7.2 million. Your chance of winning in Las Vegas are far greater than this. Yet, we believe that. That is why none of the drugs have ever worked. When they cost here and there, most of it is placebo effect. And this is the other problem that the pharma and the medical industry is stuck in. For every gene, there has to be a disease and one drug. This is the problem in medicine. We're doing the same thing again and again and expecting a different result. That's why we have so many Me Too drugs. If you look at statins, there are 10 different statins by everybody else because they're just copying each other. It's called the Me Too drugs. So we are not doing anything different. We are just doing the same thing again and again, hoping for a miracle. That's not going to happen. And this is the question that nobody ever asks. You see, you have all these drugs for every disease. The question always nobody's asked is, hey, if one of them worked, we wouldn't have, for example, diabetes. There are 120 drugs to treat diabetes. Even if one worked, the rest would be out of business. It's like our software, There are only Microsoft, there's Apple, and possibly Linux, and maybe one more. Everybody else is out of the system. They've lost. So this is the basic approach we took, that food is medicine. This is what Hippocrates said to every physician. I don't know if it's part of the Hippocratic oath, probably it's not. He who does not understand food, how can he heal the diseases of men? So the key is to understand food because we eat food to boost our immunity. That's how we live. We don't take drugs to live. So this is where, we started this whole project. There was a guy called Stephen D. Felice, the chairman of the Institute of Medicine. He must be close to 100 by now. In 1995 or 96, he gave a lecture in New York. I happened to attend. End of the lecture, he mentioned that one good nutraceutical will wipe off all the drugs. So everybody laughed as if it was a joke. But I said, what if he's right? This particular sentence stuck in my head. This was the business plan, one nutraceutical to wipe off all the drugs, because you need to be focused, and this happens to be the best way to focus on. So the question we ask is, if you have hundreds of drugs for one disease, why not one drug that will target many diseases? And you see, there is a history of this product. The history just happens to be that in the early 1880s, the Dutch physician who went to East Indies, which is now Indonesia, he found the chicken got polyneuritis when they had polished rice. But when there was nerve degeneration, paralysis, and so on. But when he put back unpolished rice, it disappeared. And this is the key. What was removed is our product. And that product is, we call it metadichol. This is what the product contains. It's a very simple molecule, just simple carbon and alcohol. And this is found in all the foods you eat every day. So the C28, the middle one, is the major one. It's over 80%. The rest are minor. This is about, this is about 85%. This is about 10%. This is about 5%. We get it from the skin of the sugar cane or the whatever is removed from the top of the rice is this product. And the problem with this product is it's a wax and it's totally insoluble in water. This is what is sold in the market. It's totally useless because never absorbed by the body. We then convert this, using nanotechnology to a product which is half the size of a human virus. This is the product which the first thing we did. This is going way back in 2009 or 10. 2009, we started a small study to see how good this is. So we took 15 patients who had metabolic syndrome and see what happens to all the parameters. So there were 7 females, 7 males, and there was one person who happened to be a guy who was not supposed to be there, but there were no diet control, no nothing. The first thing we noticed was everybody's blood pressure became normal very quickly. We're talking in around 3 weeks. Just became normal. Everybody said they had a slight headache, and then we found out that it was because their blood pressure were being normalized. Then we looked at each and every person's blood pressure, and you can see clearly it affected everybody across the board. I mean, there were people with 180 becoming 120 in just 12 weeks, which is unheard of, And if you look at the common drugs in the world, it takes about 3 years, 4 years, 5 years, and they don't even get to 120. See, the other thing that we noticed is improved lipid profiles. We see HDL go up. We see cholesterol pretty much normal. We don't touch cholesterol that much. I will tell you why. But here is the interesting thing. In week 24, there was a sudden jump of cholesterol across the board, everybody. I didn't understand for a while what was happening. 2009, August or so, or September, around that time. This turned out to be the reason why cholesterol went up was during that time, we had the avian flu, 2009. And this is where the body's defense, cholesterol shoots up. Whenever you have an infection, cholesterol goes up. And none of these people ever got the flu. So that was the first hint that it works in infectious diseases as well. The other thing that we do is something that is against medicine. People have been taught. We increase LDL. People say, wow, why do you increase LDL? See, LDL is not bad cholesterol. It's actually good cholesterol. The problem arises when the size of the LDL is small. When you make it big, the number for LDL doesn't matter. You can have 200 LDL. As long as the particles are big, it doesn't matter. It's only when they're small you have a problem. And this is what it is. You see here, the graph I have, you see triglycerides go down, and you see there is the inflection point around 1.3. The ratio of triglycerides to HDL keeps decreasing. As it decreases, the size of the LDL particle increases. So when it goes below 1.3, all the particles are big, and they're anti-inflammatory at that stage. We also have CRP levels go down, and other parties we see vitamin C go up. One of the interesting points here is our p-values are all without double-blind study or whatever. Our p-values are very good, just in open study. And just to give you CRP levels, the statin, one of the statins that is used, it took them from 0.23 to 0.19. It took them 5 years, and they considered it a great study. Here, this is actually 0.23 to 0.19. It's 0.04 reduction. We have a reduction from 3.6 all the way to 1.9, nearly 50%, 1.8 milligrams per deciliter. It's a huge number compared to this. And we also have, improvement in insulin resistance, blood sugar going down. This was in this population. One of the things I want to talk about is we are able to produce vitamin C. Now, cows produce vitamin C, but from the guinea pig, macaw, orangutan, chimpanzee, and the humans, we stopped producing human vitamin C. That is, a conversion of glucose becomes vitamin C. All the animals, people say don't give sugar to dogs. I don't think it's a risk because the dogs will convert the sugar directly back to vitamin C. So we are the only species which we can't produce vitamin C in the body. But because if you could produce, you would solve the problem of diabetes, because all the sugar will become vitamin C, you have an antioxidant, And so we did a study of vitamin C patients. There's another study we did of 30 patients. And what we saw here is a fourfold increase in vitamin C levels. This is published work. And the p-value are amazing. And once vitamin C goes up, a lot of good things begin to happen. And this is what it is. You have glucose, it goes to vitamin C. There is this enzyme called GULO, G-U-L-O, that's not active in human beings, but it's active in all the other species. The last time it was active in the human cell was about 40 million years ago. And you see here, if you somebody had done a study in the proceedings of the National Academy of Sciences, they gave a lot of vitamin C. This is up to 1,000 milligrams. The level you reach is around 75, okay? So it's a micrograms, not a whole lot. It doesn't matter how much they went from 30 milligrams of vitamin C to 1,000 milligrams of vitamin C. The level plateaus out at 74. But what happens in our case is interesting. People who have very low vitamin C, for example, this person's vitamin C was 0.15. And in a 90-day period, 1160% increase. But what happens when this increase happens, you see his fasting level comes down, his muscle mass increases, triglycerides came down, HbA1c dropped, and his body fat dropped. So a lot of things begin to happen when you produce your own body's vitamin C, which you don't get if you take it externally. Here is another case of a person who went from 13 to 107, a 900% increase. And look at his TSH levels. It was 26, dropped all the way to 4. And look at his AEC, which is absolute eosinophil count. He's an asthma patient.